introduce you to Karmella Haynes. Karmella is an assistant professor in ASU's School of Biological and Health Systems Engineering. Her group uses synthetic systems and quantitative biology to engineer useful gene and protein-based biological devices and to deepen our understanding of molecular cell biology. They operate biological devices primarily in human and mammalian cells, accelerating the pace of therapeutic technologies, such as tissue regeneration and customizable protein-based drugs, via modular design, which is a grand design that shapes her group's research plans. Dr. Haynes earned her PhD in molecular genetics at Washington University in St. Louis, Missouri. Her postdoctorate training at Davidson College and Harvard Medical School focused on designing bacterial DNA for mathematical applications and engineering synthetic proteins to control human cell fates. Dr. Haynes?
Thank you. I'm going to test is my mic on? Yep. Okay. All right. Thank you. So now we're going to get into an area that's a little bit technical. But fortunately, at least I think there's an interesting story behind all the technical information. So I'm going to start off by introducing you to a vision that I've had and something that I've been working on for the past couple of years as a bright, enthusiastic, ambitious junior faculty at ASU. So not long ago, actually exactly 3 years ago, I was living in Boston, Massachusetts, working under an established professor, Dr. Pam Silver. And her lab was a lot of fun. It was the first time in my career where I was in a place that encouraged creativity as much as it encouraged the science. So creative, in fact, and I don't have it right here for the sake of time, that we, in our free time, as we worked like crazy, we made a video and posted it on YouTube that almost went viral. It was based on Lady Gaga's Bad Romance. It was called Lab Romance. And so I was the model. I did like the strutting part of it. Anyway, so that's just a little bit of my background. And so, sometimes when I step back and I look at an idea that I sort of come up with, I say, that's ambitious. People are going to shoot that down. But, I kind of move forward anyway, and I'm stubborn enough to try to get support for it. Because to me, accomplishing that vision is pretty much more important than anything. So I'm going to give you a little bit of context. I'm going to introduce you to the problem that I'm attempting to solve with this big idea that just sort of haunted me for a while, and that I finally am sort of developing a beta version for. So sort of going back to my own profession, I'm what you call a synthetic biologist. And so this is basically genetic engineering, but with the goal of sort of pushing the envelope by putting together bits and pieces of DNA that not only allow you to express insulin in something like yeast, but allow the yeast to express insulin when they're hit with a certain wavelength of light in a certain environment, under certain conditions, in a certain temperature. The yeast may even be able to read where they're located in a certain part of the environment and then start producing insulin and then shut themselves off so that they don't overproduce and then kill the culture. So that's kind of a fictional illustration of the types of things that synthetic biology is aiming to do. And we're actually taking cues from engineers and some of the basic principles that they use to plan out building machines so that they function in a reliable way. So the field that I'm in is ambitious in and of itself because biology has this notorious reputation for being noisy, okay? I want to make that point because it's very important. And so the noisiness of biology is one of the problems that inspired this project that I'm going to tell you about. So as a synthetic biology, I take, some of the tiniest parts of life. So that's DNA that's illustrated up there. DNA molecules, you can cut them apart with enzymes, paste them together with enzymes, and sort of cobble together what we call synthetic genes. Now, there is lots and lots of DNA out there. If you've taken basic biology, you know that what makes every species unique is its unique genetic code. The genetic code is universal, but the combinations of A, T, C's, and G's will do different things depending upon what cell you put them into. So just imagine every single species that exists in the world and trying to understand what every single bit and piece of DNA does so that you can build something that functions the way you want it to, like a new type of medicine or a new type of antibiotic or something to treat a venom bite or anything like that, all of these biomedical challenges. Well, right now, sort of going back to the engineering analogy, an engineer can go to a place like Radio Shack or, wherever, like some sort of hardware store, with some particular specs in mind for the part that they want, go to the shelf, choose that part, go home, put it into their machine. There isn't anything like that exists for biology. For a biologist, what we have to do is go to some place that's kind of like a Radio Shack, a plasmid repository like Adgene or something like that, or a DNA synthesis company. And so instead of the salesperson telling me, okay, well, here's your part, this ought to work just fine, let us know how that works out, they say, oh, well, would you like to purchase a library of 10,000 DNA fragments? It'll only cost you $25,000. Something like that, right? So I'm kind of making these numbers up, but it does get very pricey. So that, I hope that helps you to understand why, if you have any friends that do research and they start talking about grants and million-dollar grants, that's where a lot of the tax money is getting sucked into. We have to buy a lot of materials to do a lot of ad hoc guesswork and try lots of different things until we figure out what works. On top of that, something that works in one lab might not work in the other. If I try to use this in bone cancer cells, it might not work in blood cancer. So there's a lot of time and money invested in sort of trying to puzzle together things from scratch every single time a lab decides to take on a project. So I'm going to move on to talk about sort of the specifics of the project that I came up with. Well, what am I going to do to try to solve this problem? Well, so I teamed up with a DNA collection curator at an organization housed at ASU called DNASU. It's a combination of DNA and ASU. I think it's a very nice name. So we were inspired to come up with a different title, SB-ASU. SB stands for Synthetic Biology at ASU. And so the analogy that I'm showing you here is pretty simple. So we chose, to fashion ourselves off of Home Depot. Okay, so those are all of the figures at the top there. So if you have a design project at home, you can go to Home Depot, choose your tools, purchase them based on the specs, on the dimensions of the thing that you need, of the types of things that you need, assemble them, put them together into a toolbox, and build your project. So that's the table on the right over there. So how is our project going to resemble Home Depot? So this is sort of the ideal scenario here. So we have bits and pieces of DNA. DNASU at ASU houses about 30,000 individual pieces of DNA that are all related to human cells in some fashion. So that's just for human cells. So just imagine the potential of sort of putting together new biomedical innovations with that. Thanks. So what we hope to do is to use the materials from DNASU and then couple them with an online information repository called Figshare, and I'll tell you why that's important in a minute. So then once we put this repository together, researchers from all over the globe will be able to access this. So this isn't only just for scientists at ASU or in Phoenix or in Arizona. This is meant to be a global resource. And so just imagine all of those labs working in parallel, sort of exchanging information with each other. So that's the key part. That's a key part of the project, is that when you get material from the repository, you also have to give, right? Once you test that DNA fragment in your cells, let's say someone in Utah is working on bone cancer, they test part number 54018, they record a certain level of function, they dump that information back into the repository. 5 other labs across the country are also testing the same part, because these parts are interchangeable and modular, so people will tend to reuse the same things. So any of that data that comes out of the reuse starts to build out the information for a single part. So kind of imagine Yelp. in this regard, right? Yelp is kind of serving, it's acting like a Wikipedia of restaurants because people are sort of collaboratively assessing the quality of that restaurant, and now you have this rich resource of information for a single restaurant, for lots of restaurants in the case of Yelp. Okay, so yeah, we're aiming to make this a fresh, holistic, and potentially disruptive approach to overcome critical hurdles in synthetic biology. And I say disruptive because the typical approach is that, okay, well, you know, a smart person, PhD, establishes their lab, they get a 30-person army in there, they apply for $1 million grants, and then they just solve the problem, you know, brute force, and it's an isolated effort. They're competing with powerhouse labs all over the country. That is not our approach, okay? I believe that science, or at least biomedical engineering and molecular biology, are broken in that way, okay? Sure, you know, enough focused effort from one very talented lab might discover some really neat things, but in terms of solving the toughest problems and actually making medicines and solutions, we're very, very, very bad at it. Case in point, if you follow synthetic biology news, one of my favorite synthetic biologists ever, Jay Kiesling, he developed an anti-malarial drug that could be produced in yeast instead of a very rare plant that's grown in West Africa. So that was a huge accomplishment for synthetic biology, but by the time they got that to work, they had spent millions of dollars and 10 years developing that. Just imagine if lots of labs were talking to each other and informing each other what works and what doesn't, so that you wouldn't have to reinvent the wheel and just trudge forward by yourself. That solution could have come along a lot faster, at least I believe so. Okay, so this is just an overview of the folks who will benefit, not only research labs, but students. So this is going to be an online resource, and just imagine that, you know, a biomedical or a budding biomedical engineer really wants to understand, you know, how feasible is building a synthetic gene circuit that kills cancer? How does that work? This information is going to be accessible to everyone. And that's me up there in the picture teaching Biomedical Engineering 100, so Intro to BME. And then so you bet that because I'm going to be in charge of this database, I'm definitely going to be exposing our 200 new admits per year to this resource into synthetic biology. Okay, so this slide, I don't want to emphasize the details, but this is just to show you that this is happening. As of March 14th, we were informed that we received support, financial support, from a wonderful organization called Women in Philanthropy. And then so this is especially touching to me because I ran across a quote from someone named Dr. Peggy Rissmiller, and she's referencing a book called The Woman on the Mountain, I believe, if I've got that title correct. And I ran across this quote that really struck a chord. She says that women often have a more holistic and unconventional approach to science that your typical go-to funding agencies don't always appreciate. And then so, when I got that letter that I had received $75,000 to kickstart this project, I just wanted to shout hallelujah. That's awesome. Okay, so my colleague Catherine Saylor and I are ready to get started this summer building up this database. Of course, there are some obvious hurdles, issues with data normalization. How do you take data out of one lab and match it up with data from another? We're going to collaborate to work on those details, but we're aiming to get the beta version of this launched by early next year. Okay, and so with that, I'll thank you for listening, and I look forward to your questions at the end. Thanks.